In last class, in this chapter, human health and disease, and this chapter, the last class, I discussed about immune system in the body. Last class, I have discussed about immune system in the body. Now, I have discussed about AIDS. AIDS means AIDS stands for acquired. AIDS stands for acquired immunodeficiency. Acquired immunodeficiency syndrome. In this course, I will discuss about AIDS. AIDS, the word AIDS stands for acquired immunodeficiency syndrome. Acquired immunodeficiency syndrome. It means deficiency of. It means deficiency of immune system. Deficiency of immune system. Deficiency of immune system. Then syndrome means group of symptoms. Group of symptoms. Syndrome means group of symptoms. And the AIDS was first discovered. Acquired immunodeficiency syndrome was first discovered in 1981. AIDS, this syndrome or disease was first discovered in 1981 in USA. In USA, among homosexuals. Among homosexuals. And next point. The AIDS, this syndrome, this disease was caused by HIV virus. AIDS, the syndrome or the disease was caused by HIV virus. HIV stands for human immunodeficiency virus. Human immunodeficiency virus. Next, HIV virus is grouped into RNA virus. So this HIV is a RNA virus. HIV virus is a RNA virus because RNA acts as a genetic material. And the HIV is a RNA virus and it is grouped under retroviruses. It is HIV virus is grouped under retroviruses. Means retroviruses. What are retroviruses? Why RNA viruses is grouped under retroviruses? Retroviruses, RNA virus, HIV is a RNA virus. Then RNA viruses are grouped under retroviruses because the retroviruses convert RNA into DNA. RNA into DNA. by using the enzyme reverse transcriptase. Reverse transcriptase. Reverse transcriptase enzyme. Normally, so RNA in retroviruses or group of viruses, in these retroviruses, RNA converted into DNA. This RNA synthesizes DNA in the presence of a viral enzyme called reverse transcriptase. Normally or generally, so DNA synthesize RNA. DNA synthesize RNA. So this is called transcription. This process is called transcription. This process is called transcription. And this virus, HIV virus, specifically attacks T helper cells. HIV virus specifically attacks helper T cells, T lymphocytes. So HIV viruses specifically attacks helper T cells. Clear? It's about introduction about AIDS. AIDS, the word stands for acquired immunodeficiency syndrome. This means deficiency of immune system because the HIV virus particularly attacks T helper cells. As a result of this, the number of T helper cells gradually decreases. Because of this, our immune system is collapsed or destroyed gradually. That's why it is called deficiency of immune system. So syndrome means what? What is syndrome? Syndrome is a group of symptoms. Syndrome means a group of symptoms. Then AIDS was first discovered in 1981 in USA among homosexuals. Then HIV, AIDS was caused by HIV virus. HIV, the word stands for human immunodeficiency virus. Human immunodeficiency virus. So HIV virus is an RNA virus. HIV is an RNA virus because the HIV virus has RNA as the genetic material. HIV has RNA as the genetic material, or in HIV virus, RNA is the genetic material. That's why it is called RNA virus. And HIV virus is grouped into the retroviruses, grouped under retroviruses, because the HIV virus, the genetic material RNA, this RNA, genetic material of HIV, converts RNA into DNA. RNA synthesize DNA. Convert means viral HIV RNA convert into DNA or produce DNA. In the presence of reverse transcriptase, RNA convert into DNA. In the presence of reverse transcriptase. That's why these RNAs are called retroviruses. Normally, DNA can synthesize RNA. Normally, in normal process, DNA synthesizes RNA. This process is called transcription. Reverse transcription means RNA synthesize DNA in the presence of a viral enzyme called reverse transcriptase. Next, that's why it's called reverse transcription. This process is called reverse transcription. Then, HIV virus specifically attacks helper T cells. Attacks helper T cells. So, next, a structure of HIV virus. Structure of HIV virus. Spectrum of HIV virus. Glycoprotein spike. Glycoprotein spikes. Lipid bilayer. Lipid bilayer. Protein coat. Protein coat. RNA filaments. RNA filaments acts as genetic material. Acts as genetic material. And then reverse transcriptase. Reverse transcriptase enzyme. Reverse transcriptase enzyme. Reverse transcriptase enzyme. Reverse transcriptase enzyme. This is a diagram of HIV virus. HIV virus. See, HIV virus is a spherical shape. HIV virus is a spherical shape. Next, it has 90 to 120 nm in diameter, shape as a spherical in diameter about 90 to 120 nanometer. shape 90 to 120 nm diameter. Then HIV virus consists of this see the diagram. HIV virus consists of two DNA filaments. Two DNA filaments act as a genetic material. RNA HIV virus contains two RNA filaments. RNA filaments. HIV virus contains two RNA filaments. These two RNA filaments act as genetic material. Acts as a genetic material. Next. These RNA filaments generally associated with the enzyme called reverse transcriptase. Reverse transcriptase enzyme. Reverse transcriptase enzyme. Reverse transcriptase enzyme. See, reverse transcriptase enzyme. Reverse transcriptase. The HIV virus is covered on outer and inner protein coats. Protein coats. The HIV virus is covered by outer and inner protein coats. HIV virus is covered by outer and inner protein coats. Clear? So HIV is a spherical shape and it measures about 90 to 120 nanometer in diameter. Then HIV virus has two RNA filaments in the center of HIV, the core, in the center or core, two RNA filaments are present. These two RNA filaments acts as a genetic material. So then these two RNA filaments are associated with an enzyme called reverse transcriptase enzyme. And the RNA, so virus is coated or covered by outer and inner protein coat. And next point, the above the protein coat, above the protein coat, another envelope or another membrane is present. It is called lipid bilayer. So, above the protein coat, above the protein coat, another envelope or membrane is present. It is called a lipid bilayer. It is called lipid bilayer. It is called lipid bilayer. From the lipid bilayer, the glycoprotein, viral glycoprotein spikes, from lipid bilayer, viral glycoprotein spikes are projecting and these viral glycoprotein spikes as a receptors form T helper cells. These glycoprotein spikes present on the protein layer. These glycoproteins are very important because these glycoprotein spikes as a receptors specifically for T helper cells and enabling these glycoprotein spikes enabling the entry of virus into the host cell, spherical shape. It is about 90 to 120 nanometer in diameter. HIV virus has two RNA filaments. These two RNA filaments act as a genetic material. These RNA filaments are associated with viral enzyme called reverse transcriptase, and the HIV virus is covered by outer and inner protein coat. Above the protein coat, the another layer is present. It is called lipid barrier. From the lipid barrier, the viral glycoprotein spikes are projecting out, and these glycoprotein spikes have receptors specifically for NPER T cells, and these glycoprotein spikes ends to entry of viruses into the host cell. This is the structure of the HIV virus. Next, multiplication of HIV virus. So once the HIV virus enters into the host cell, how it multiplies. This is the next concept. How the HIV virus is multiplied inside the host. Next is multiplication of HIV virus. So multiplication of HIV virus. Next concept. Multiplication of HIV virus. 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 Retrovirus. 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 So virus infected a normal cell. Virus infected the normal cell. Virus infected the normal cell. So next. Virus infected normal cell. It's an animal cell. Animal cell. This is plasma membrane. Animal cell, plasma membrane. Plasma membrane. Plasma membrane. Viral RNA. Viral RNA introduced into introduced into cell. Viral RNA introduced into the cell. Then viral RNA incorporated into viral DNA incorporated into host genome. Viral DNA into host cell. Incorporated into host cell. Then nucleus. So this is nucleus. This is DNA, host DNA. Nucleus and the host DNA. Viral DNA is produced. Viral DNA is produced by reverse transcriptase enzyme. Viral DNA is produced by reverse transcriptase enzyme. These are the new viruses are produced. The new viruses infect other cells. The other cells. This is so multiplication of HIV virus. This is about the multiplication of HIV virus. Multiplication of HIV virus. See, how exactly the HIV virus can multiply in the host cell? What's the mechanism? What exactly happens during the multiplication of HIV virus? See, this is a retrovirus. Virus infected the normal cell. We know already what is retroviruses. Retroviruses are the group of viruses. They convert RNA into DNA in the presence of reverse transcriptase enzyme. So then these retroviruses infect the normal cell. See, this is the host cell or animal cell, plasma membrane. So the retroviruses, that is viral RNA, entered into the cell. So then this viral RNA enters into the cell, it produces viral DNA. Then after production of the viral DNA in the presence of reverse transcriptase, this viral DNA incorporated into the host cell, nucleus and DNA. So then it results in the production of new viral particles. Then these new viral particles release and infect other cells. See, very first, when the virus entered into the body, it attacks helper T cells. Helper T cells means macrophages. Macrophages infects the macrophages of immune system. Clear? Very first, the HIV virus enters into the host body. Within the host body, the virus specifically attacks helper T cells or T lymphocytes called macrophages of our immune system. Specifically attacks helper T cells of our immune system. Next, what happens in next step? So the viral RNA is converted into DNA copy. Viral RNA is converted into DNA copy in the presence of an enzyme, reverse transcriptase. So the viral RNA converted into DNA copy in the presence of reverse transcriptase. Next step, the viral DNA. The viral DNA incorporates or entered into the host cells. DNA. In the third step, the viral DNA incorporated into host cells DNA and directs the infected cells to produce virus. So viral DNA enters into the host cells and directs the infected cells to produce the virus. So next step, macrophages simultaneously the macrophages, these viral particles attacks helper T cells. Simultaneously, these viral particles can attack helper T cells, and the macrophages acts as a HIV factor. Simultaneously, the viral particles can attack T cells or T lymphocytes or macrophages. So the HIVs, in this way, the macrophages acts as a HIV factor. Then these HIV viral particles produced replicates and produce new progeny viral particles. Clear? Simultaneously, the T cells, these viral particles attack T cells, helper T cells means T lymphocytes. As a result, the macrophages are considered as a HIV factor because all the viral particles are multiplied, replication, and it produces new protein. That's why the macrophages are considered as a HIV factor, because in the macrophages the HIV virus particles are multiplied, replicated and produces new progeny viral particles. Clear? Listen once again. Viruses, HIV viruses are entered into our body. Specifically, they attack helper T cells of our immune system. The viral RNA next get converted into DNA copy in the presence of reverse transcriptase enzyme. This process is called reverse transcription. This process is called reverse transcription in the presence of a viral enzyme called reverse transcriptase. Then viral DNA enters into the host cell's DNA and directs the infected cells to produce viral particles. Then the viral particles within the macrophages multiply, replicated and produce new progeny of viral particles. That's why the macrophages, that's why the macrophages are called HIV factory. So because in the macrophages, the viral particles can replicate it, multiply and produce new progeny. Because of this, the macrophages are called HIV factory. These new viral progeny, the new viral progeny is produced. The new viral progeny is produced and released into the blood and in the blood they infect other helper T cells. Infect other helper T cells. Clear? The new viral progeny are produced in the macrophages, then released into the blood, and in the blood these new viral progeny are attack other helper T cells within the blood. This process is continued or this process is repeated. This process is repeated, leading to progressive decrease in the number of progressive disease in the number of T cells or T lymphocytes in the body of the infected person. Gradually, in the body of the infected person or HIV patient, the number of T lymphocytes are decreased. Because the new viral progeny can attack these T lymphocytes in the blood. As a result of this, the number of T cells or lymphocytes goes on decreases in the body of the infected person. Clear? Because this process is repeated, leading to progressive decrease in the number of T cells in the body of the infected person. So ultimately, in the infected persons, the number of T lymphocytes goes on decreases. In this period, the patient suffering from botus of fever, diarrhea, and weight loss, In this stage or in this period, the HIV patient suffering from botus of fever, diarrhea and weight loss. So this process goes on continuously. Gradually, the number of T lymphocytes decreases. As a result of this, the patient infected with other infections like mycobacterium, viral diseases or infections, then fungal diseases or infection, and the parasite infection by toxoplasma, See, this process is continuous. Gradually, the number of T lymphocytes goes on decreases. In this stage, the AIDS patient or the infected person cannot tolerate infection or does not produce defense against the other infections like mycobacterium, viral diseases, fungal diseases, and even parasitic disease caused by toxoplasma. It means the HIV patient cannot shows immunity or resistant. The AIDS patient does not defense. The AIDS patient immunity system does not protect against the diseases caused by mycobacterium, viral diseases, and fungal diseases, and parasitic diseases like toxoplasma. So then this is because the AIDS is called as a syndrome, not a disease, because all the diseases symptoms, mycobacterium disease, viral disease, fungal disease, and toxoplasma, all these symptoms occurs in the HIV patients. That's why HIV is called as a syndrome. It is not a disease. Clear? Because the HIV patient does not protect from mycobacterium, viral, fungal, and toxoplasma. So the patient becomes so immunodeficient. The AIDS patient become so immunodeficient. Next, the AIDS was diagnosed clinically by ELISA. AIDS clinically diagnosed by ELISA. ELISA stands for enzyme linked immunosorbent assay. Enzyme linked immunosorbent assay. ELISA is a diagnostic test against the HIV or AIDS patient. Enzyme linked immunosorbent assay. And also Western blotting is also used for diagnosing the HIV or AIDS. Clear? So initially the AIDS patient shows borders of fever, diarrhea, weight loss, and gradually the patient, aged patient, does not resist against infection of mycobacterium, viral, fungal, and toxoplasma. So the AIDS patients become so immunodeficient. Then this HIV or AIDS was clinically diagnosed by ELISA, enzyme linked immunosorbent assay, and Western blotting techniques. Western blotting techniques. AIDS was diagnosed. Here, this is about multiplication of HIV virus. First, HIV virus can enter into the body, host body, and specifically attacks helper T cells. Then, the viral RNA convert DNA copy in the presence of reverse transcriptase enzyme. Then, once the DNA copy is produced, this DNA, viral DNA, can incorporate into the host cell's DNA and directs the host DNA cells to produce virus or viral particles. So simultaneously in the macrophages, the viral particles are produced and macrophages acts as a HIV factory. Because the macrophages continuously produces progeny viral particles. Okay? And in macrophages, what happens? The progeny viruses are multiplied, replicated, and so produced more viral progeny. So multiplication, replication of progeny viruses. Finally, from macrophages, the progeny viruses are released into the blood, and in the blood, these progeny viruses attack other new helper T cells, other T helper cells in the blood. So these processes are repeated, leading to progressive decrease in the number of T lymphocytes or helper T cells. In the body of the infected patient or infected person. So in this stage, the patient suffering from bouts of fever, weight loss, and so diarrhea. This process goes on continuously. As a result, gradually the number of T lymphocytes further decreases. Then this time the patient cannot tolerate the infection of the mycobacterium, viral infection, fungal infection, and infections of parasites like toxoplasma. Because of this, the patient becomes so immunodeficient. So that the AIDS patient shows symptoms of all these infections. That's why AIDS is called as a syndrome, not a disease. So next, the AIDS is diagnosed clinically by a test. It's called ELISA. ELISA means enzyme linked immunosorbent assay and Western blotting techniques. These two techniques, ELISA and Western blotting techniques, are generally used to diagnose the AIDS.